You know what? Maybe I'm going to turn closed captionings off unless people want it. I forgot because I had some students last year who were hard of hearing, and so they needed it on. But it also just distracts me. But if anybody wants it back on, let me know. So we're going to talk about seed dormancy and ecology. But the first thing we want to talk about is what a seed is and why most plants have them. So here in my picture, I have some seedless plants. So we have bryophytes, and so they're seedless and they're non-vascular, so they don't have the ability to transport water through the plant, and they reproduce by spores. And then next to it, we have a fern. These are seedless vascular plants, so they have roots, they have the ability to transport water, but they still just reproduce by spores. So both of these groups are dependent on water for reproduction. But, you know, if you're living in an area where water is not super abundant, that's not necessarily the best strategy, or if water is very seasonal. So I grew up in Utah, which is a desert, and then I did graduate school in southern Arizona, which is also a desert. And so rainfall is very, very seasonal there. And so it was nice to have a way for the plant to store the next generation and then wait for conditions to be favorable to actually grow a new plant. And so gymnosperms, so pines and their relatives, were the first ones to come up with that. And then the angiosperms, or flowering plants, perfected that. And I will try and keep an eye on the chat. Oh, I'll say I don't think that one was for me, in case there are questions. So the great thing about seeds is that they protect the embryonic plant, and they basically give them the ability to wait until conditions are favorable to grow. I hate it when this happens. Okay. So this is a pine seed. So you can see the pine cone here. And so I just want to give you like a general version of what a seed looks like. So in a seed, you're going to have the embryonic plant, and then you're going to have a food supply. And that food supply is there because when the seed germinates, it needs something to fuel that growth until it can start to photosynthesize. And then they're going to have a seed coat, and this is going to cover it so that nothing can get in to get to the seed. So it makes it a lot harder for fungi or other environmental stresses to come in and kill the embryo before it's ready to grow. So the structure of the seed is specifically there for embryo protection and for the embryo nourishment. So we're going to look at two different types of seeds structures for the most common types of plants. So angiosperms are the most common. So that's going to be your flowering plants. And we have two groups. We have monocots, which are the things in the grass family. So think about corn and maize, or wheat, rice, all the grasses you see outside. So they're called monocots because they have one cotyledon. So a cotyledon is the seed leaf. So that's that first thing that sometimes we'll see pop out. Sometimes it stays underground. They store their energy reserves in an endosperm. And so when we eat grains, that's what we're eating, is we're eating that starchy food reserve for the embryo. In the monocots, they have a coleoptile. This is what's going to push up through the soil. And then we have the coleorhiza, which is the seed root. So that's going to be the first part that comes out during germination. And then we have a seed coat. So with corn, that's actually fused to the fruit. So fun random facts. I'll like to intersperse them occasionally. And that is for protection. It prevents water uptake, and it also prevents the uptake of oxygen which is one of the things that the plant needs to germinate, because without oxygen, we're not going to undergo cellular respiration. So we're not going to make ATP, which is that energy that we need. So dicots are the example dicot I have here is a legume. So this looks like a bean seed. So think of things that basically, if they're not like lilies or grasses or things like that, that don't have like long straight leaves, then they're probably dicots. And so here, instead of having an endosperm, well, so first of all, they have two cotyledons, so they have two seed leaves. And instead of having an endosperm, their energy reserves are actually absorbed into the cotyledons. And so when you break open a bean seed, what you're basically seeing is those seed leaves, and they're very, very large. So the first part of the plant that comes up is called the epicotyl. So that's going to be your seedling stem. Then we have, sorry, forgot. So this is how they emerge. So we have epicotyl is where the cotyledons come up first, and hypocotyl is when the stem comes up first. And then the radicle is what we call the embryonic root. So both of them have the same structure, and then we have the seed coat that surrounds and protects the seed. So sometimes people will also refer to the seed coat as the testa. So if you were to hear that, you would just know that they're the same thing. So I'm going to go through the basic steps of germination, so then we can talk more about what happens with dormancy, because that's kind of the fun part. So all seeds, no matter what, the first thing they have to do before the embryo starts to grow is that they have to take up water. So this process is called imbibition. Then we're going to break that seed coat. So when the seed coat breaks, then we're going to be able to have oxygen come in, so the cellular respiration starts. So now we start breaking down starches and sugars to make ATP, so that energy that's going to be needed to help the new plant grow. So when we start breaking that stuff down, we call that mobilizing the reserves. So for most plants, what we have is starch. And so that starch is going to be broken down into glucose, which will then be broken down further to create energy. Then that, once you have energy and you have resources, cells start to divide. And then we have the emergence of the radicle, or that embryonic root. And that actually is when plant biologists say that germination is done. We're like, okay, now germination is officially occurred when that seedling root has emerged. And so here's just a picture where you can see, this is an alfalfa seed, where you can see the seed coat has broken a little bit. And we can actually see the radicle coming out. And usually, so you can kind of see when something's about to germinate if you're looking at it under a microscope, but it's always really amazing like how quickly that seed root will grow. And so here we can see, usually we consider it completely germinated when that radicle is about the length of the seed. And you might be looking at this picture and you'd be like, why is there a picture, just an up close picture of the root? Well, it's because there's actually a hormone called abscisic acid, or ABA, that can prevent the process of germination occurring. It's actually its job. And so what they had done in this experiment was add ABA to show, hey, that it stops the process of germination. So we have a bunch of hormones that are important in seed development and germination. So we have a hormone called cytokinin, and cytokinin is really, really important in cell division. So it's really important in making the embryo, and then when it's time for the seed to germinate, cytokinin is really crucial for cell division to occur to help the plant get bigger. Then we have gibberellin, and gibberellin is what controls this process of starting the breakdown of starch. So over in this picture here, water uptake stimulates gibberellic acid to go into this living layer of cells on the outside of the seed called the alurone. Then the alurone cells tell the endosperm to start breaking down. So they release some enzymes that break down the starches in the endosperm, and then that fuels seedling growth. And we have auxin, and auxin works together with cytokinin to make sure that cell division occurs correctly. And without auxin, the cells won't actually get any bigger. So it's really important in plants that if you don't have auxin, you're not going to get, your cells will just get really, really small and they won't get any bigger. And then abscisic acid, like I said in the last slide, is really important in making sure that the seeds stay dormant and they don't germinate until they're supposed to. And abscisic acid also is really important with seeds because seeds can go down to about 10% water content. And so think about yourself, and if you went down to 10% water content, you would be dead. And a plant that went down to 10% water content would also be dead. And so abscisic acid actually helps seeds stay in this super, super dehydrated state and still have everything protected so that when water does come, they work the way, all those cells and structures work the way they're supposed to. So it's pretty cool. And it's funny because abscisic acid, people named it that because they thought it was important in abscission, which is when leaves or fruits fall off of trees. And it's not involved in that at all, but they never changed the name because so it's one of those things where the name actually has no relationship to what it does. So we can actually see an example of what happens if abscisic acid is removed or disappears early. We have something called viviparity. And this is when a seed germinates in a completely inappropriate place. So here we have a cob of corn, and we have some corn seeds that have already started the germination process. It's very warm in Arizona, obviously. And my in-laws, if they store tomatoes on the counter for a couple of days, if you cut those open, you're actually going to see the tomato seed germinating inside of the tomato. And I've had students who pulled up or like opened up apples and seeing the apple seeds germinating inside of the apple. And so usually something has gone wrong. Abscisic acid either was gotten rid of early or it wasn't formed correctly. And so germination is occurring when it shouldn't. And so that would be something where dormancy isn't staying in place, which kind of leads us to what we're going to be doing next. So the reason why we're here is to talk about seed dormancy. So this is its specific scientific definition, which is the failure of a viable seed. That means that the embryo is living. So you can actually cut seeds open and stain them. And if they stay in a certain color, we know that that embryo is totally viable. To germinate when given favorable conditions. So that's usually going to be the correct temperature and water. So if you have a seed that doesn't germinate under those conditions, then we have dormancy. So we have two types of dormancy. And the first type is mechanical dormancy, and it's specifically related to the seed coat or to the seed coat. And so one is like the actual physical seed coat itself. And then sometimes the seed coat has chemicals in them that act as inhibitors to germination. And we'll go through each of these a little bit more closely. And then we have something called endogenous dormancy. And this is something that is inherent to the embryo itself. So something about the embryo is preventing germination. So we're going to go through each of these, and then we'll talk about which category they fall under. So scarification is a way that we can break dormancy. And this is something where we break the seed coat, and we'll look at each of these in a little bit more detail in a second. Then we have something called embryo maturity, so after ripening. So this is going to be endogenous. Then we have leaching, so adding water to remove inhibitors. Stratification, which is a cold temperature treatment. And that is going to be more of an endogenous thing. So scarification and leaching are mechanical, related to mechanical dormancy. Embryo maturity and stratification are related to endogenous dormancy. Like I said, all seeds need water uptake and optimal temperatures, and some seeds might need light and some seeds might not in order to germinate correctly. So we're going to go kind of dive deeper into this. And if anybody has any questions, I don't have you guys all up on the screen because it gets kind of distracting, but you can totally throw stuff in the chat and I will try and answer questions, but there'll be time at the end as well. Okay, so I put graphs in here. I wasn't sure how technical you guys like to be, but I love graphs a ton. It's one of my favorite things. So we're going to look specifically at seed scarification. So the seed coat, like I said, it acts as a barrier for the uptake of water and of oxygen. And so sometimes the seed coat was really, really, really thick. And so if the seed coat's really thick, it's going to be really hard for water and oxygen to get there. And so we actually have to physically break down the seed coat in such a way that water and oxygen can start to be taken up. So if you're a plant physiologist or biologist and you want to do this, you don't do anything fancy. You take some really fine grit sandpaper, you put your seeds down on top of the sandpaper, and then you take the sandpaper on top and you just rub it really lightly, and that's usually sufficient. But in nature, we don't have, sandpaper. We have nature's sandpaper. So most seeds, when they're dropped, or a lot of seeds, when they're dropped, they end up in the soil. So they can get buried. And if anybody spent a lot of time putting your hands in the soil, it can do a number on your hands if you don't have gloves on. And so if we think about, as the soil moves, especially if we have freeze-thaw cycles, which will cause the soil to expand and contract, those seeds are actually being rubbed back and forth by the soil. And so as they go through these freeze-thaw cycles or anything else that would move them through the soil, that's going to scratch up the seed surface. So another thing that can happen, and I'm sure you guys have noticed this, is we see lots of birds and other animals, they come and they eat fruit off of plants, and they will eat the fruits, but usually what happens is that instead of digesting the seeds get pooped out. And we know that the digestive juices are very, very strong. And so that the chemicals that are found in our digestive juices, the acids and bases, actually can break down the seed coat as well. And so some seeds won't germinate unless they have actually gone through the digestive tract. So let's just take a look at our graph here. So this is a graph looking at the effects of scarification and seed coat removal. So I can only imagine a graduate student sitting there peeling the seed coat off. Fun times, keeps everybody out of trouble. And they're looking at germination frequency. So we're looking specifically at, so intact seeds are the circles. So I don't know if you can see them here. I'm going to see if I can. Oh, let's see. We'll do the laser pointer. Okay. So the circles are here. So these are the intact seeds. So being intact, it seems to take about 10 to 12 days before we even start seeing any germination at all. And then by about 20 days after sowing, all the seeds have germinated. The triangles are seeds that have been scarified. So we've just broken the seed coat. And you can see that at 10 days, we have about 20% germination. And so it seems to make germination a lot faster, just to break that down. And then we have the ones where the seed coat's been completely removed. Germination is happens about 6 days earlier, and we complete germination a lot faster. And so we can see how the seed coat is actually really important in maintaining dormancy. Okay, so now we're going to look at specifically how some hormones and age of the embryo can impact whether or not a seed germinates. So this is a graph that is crazy. My students hate it. It's fun times. So non-photodormancy just basically means that the seed has the ability to germinate in the dark. And why they don't say that, I don't know, because we're scientists. We like to use fancy words for things. So what the researchers did here is they basically looked at, okay, first of all, so let's just look at our fresh seed. So we're just going to look at at 0 gibberellic acid being added. And so the open circles are our youngest seeds, and the upside down triangles are the oldest seeds. And we can see that just having the seeds be older, so this is called after ripening, increases the amount of germination that occurs. So what's happening here is that, remember, we said the embryo is living. So even after they fall off to the maternal plant, a lot of times that embryo is still undergoing developmental changes. And until those developmental changes are done, they're not really ready to germinate. So they're still growing, they're just doing it in a really slow and invisible type of fashion because we don't see it. But we can overcome at least some of this, or we actually can completely overcome this after ripening time period requirement by adding enough gibberellic acid, because gibberellic acid stimulates all those processes that mobilize the endosperm so that it provides the resources for the embryo to grow. So this is, like I said, it's kind of a crazy graph, but it's really cool that you can see, okay, here's a lot of different things that are acting together that can make it so that the seeds germinate correctly. So the next thing we're going to talk about is the second mechanical dormancy related thing, which was the chemicals that are found inside the seeds. So a lot of plants produce chemicals that prevent germination or the emergence of other plants. And this process is called allelopathy. And so what happens with seeds that have inhibitors in their seed coat is those chemicals are sitting there, and as they're exposed to rainfall, and so water starts going through the soil, it washes those inhibitors out. And so what's happening is that the seed is being released from its inhibition, so it can germinate, but it's put all the inhibitors out there so that its competitors from other species can germinate. And so it's kind of a cool setup so that they basically can induce kind of a dormant state on their competitors so that they basically get a head start with germination and have a better chance of outcompeting other species. So I've always thought that that one was a pretty cool system. And then there are other parts of plants that can be allelopathic. So in this picture, we have straw mulch is acting on weeds and preventing their seed germination or emergence. Okay, so the next one is seed stratification. So this also has to do with the embryo. And so we have a combination of moisture and appropriate temperature to break dormancy. And so the seeds have to be wet for this to work. So if you were to just throw seeds into your refrigerator, seeds that had required stratification, but they weren't wet, and then you tried to plant them, it wouldn't work. And so you do have to have moisture associated with this. And it's usually, if you're doing it with seeds that you have, it's going to be associated with the overwintering conditions that plant would already have. And it's most often cold temperature exposure, although in the tropics you can still have stratification. It's just you need a certain exposure to the kind of the warm temperatures and fluctuation. But in Maryland, stratification is cold. And so one of the reasons why plants do this, I mean, it makes a lot of sense, is that if you have a nice warm day in December or January and your seeds all germinate, and we all know what happens is because like right before, a couple weeks before Christmas, it was really nice. And then Christmas Day, it was cold. And so if you are a seed and you germinated, you're going to die. And so they actually have a cold temperature requirement so that it's like having a cold temperature clock. So, hey, I've been exposed to cold temperatures for long enough that when you start getting warm stretches of warm weather and warm temperatures in the soil, it's actually appropriate for germination to occur. And so it's kind of a cool setup, because plants that don't do this haven't survived because all their seeds died, so they didn't reproduce and pass their genes on to the next generation. So, like I said, I like graphs, so you guys get lots of them. So this is a really cool set of graphs that looks at stratification period, so this cold temperature exposure, and what it's actually doing to the hormones in the plant. So in figure A, the stratification period, they start with 15 degrees Celsius for 16 weeks, and then they go down to 3 degrees Celsius for another 32. So 15, I mean, honestly, that's kind of one of my favorite temperatures. Pretty nice outside. 3 degrees, we're getting close to freezing. And then germination, we make that switch from 3 degrees to 25 degrees, which is about room temperature, about 72. And we're going to look specifically at this line here in the embryo. So this is the abscisic acid, which, if you remember, is that hormone that is specifically there to prevent germination. So it helps to maintain dormancy. And if we look at the amount of abscisic acid in the embryo, as it's exposed to the stratification period, it starts to go down. At the same time, we have all this weirdness going on with gibberellic acid. And gibberellic acid is kind of a fun hormone because there's I'm trying to think. There's at least 25 different gibberellic acids, and most of them don't do anything. Most of them are inactive. So here we're looking, so we have this inactive precursor, and amount in the middle of the stratification period, this inactive precursor kind of spikes. And right after it spikes, we see a spike in the active gibberellic acid, and it's spiking specifically in the endosperm. So it's getting ready for germination. So it's getting ready to mobilize those reserves. So there's a lot of things actually happening in the seed, which we don't even really notice because we don't see anything until that we see that seedling pop out of the soil. And then we have a spike in third gibberellic GA3 in the embryo right as germination is occurring. And that helps with stem elongation. Okay, let's take a look. I have a question in the, sometimes the chat will let me see. Okay, so it says, can you please explain the process of double dormancies where some seeds like ephemerals need cold moist stratification followed by warm? I think moist stratification, then second cold moist stratification. Well, that one, that's something I don't know a ton about, but I do know that some seeds you need they require a much longer stratification period, and they need a lot of temperature variation. So I think, Marlene, that you explained this really well. So yeah, and I'm not sure why that happens. It could be, so if they're ephemerals, they might need to be like, okay, yeah, I've gotten this cold temperature, I've gotten warm, then I've gotten cold again. So next time I get warm, it would be good to go. But other than that, I don't really know a ton about it. So I'm sorry, I can't say more. Okay. I'll say, oh, sweet. And then during this whole stratification period, the embryo is actually getting bigger, which is pretty cool. So some seeds also need germination or light to germinate. And for seeds that need light, they're called positively photoblastic, and they require exposure to red light. So these are going to be seeds that are found really close to the surface. So lettuce or like basil, usually you just, like if you bury them, it's like 1/8 of an inch of soil, or you just press them into the soil surface. That's because they require light, because their endosperm is really, really small. And so if they start germinating deep in the soil, they're going to starve before they ever get to sunlight and start to photosynthesize. But other seeds, like big seeds, actually are negatively photoblastic, and they will not germinate if they're exposed to light. And this has to do because if you're a big seed, animals want to eat you. And so it's more advantageous to be buried deeper in the soil. And then you can use your bigger endosperm to make it to the surface, and your chance of surviving before being eaten is actually a lot higher. And then once the seed's ready, we can kind of just skip the seed dispersal, because I am talking a lot and I'm not through everything that I wanted to go through. So why is knowing stuff about dormancy important? Well, some of it is, if you're wanting to grow something, you need to know like what you need to do to grow it. So like if you want to grow milkweed, you need to have some stratification, or you're not going to be able to grow it. So I always look like on the back of my seed packages when I'm getting ready to plant. I'll be like, okay, what is the planting condition, so that I know what's going to be the best way to get them to grow. And that's going to be really important with native species, because they're not like seed packages that you buy in the store, where we basically bred those plants to germinate really, really easily. And so if you're trying to increase the number of native plants, it's going to probably be a little bit harder to grow those than stuff you just buy at the store. Unfortunately, things that are really important or easy to grow without any help at all are weeds. And the weeds that are really important to Maryland's ecology are invasive species. So weed is just typically a wild plant that's growing where we don't want it. So weeds are fast growing, so they're going to be good competitors. So they compete for space, nutrients, and water. So here I stole from the a talk I gave back in June on weeds, but it can be native plants as well. They can tolerate lots of environmental conditions. So weeds are they really are kind of super competitors, and they produce lots of seeds. And native weeds, like I don't like poison ivy growing in my yard. So in my yard, it's a weed, but it's native. So, and I know it has a purpose, so I'll let it grow other places. But what we worry about are invasive and noxious weeds. So these are plants that are not native to Maryland, and they are can actually outcompete our native plants. So noxious weeds are designated by government authority as injurious to health, agriculture, and the environment. So giant hogweed is one. I've never seen one in person. If you ever do, don't touch it. It will sensitize your skin. And so then if you're exposed to sun, you'll get like the worst sunburn of your life. So it's really, really dangerous. But most weeds aren't considered noxious weeds. What we are dealing with more in Maryland are invasive plants. So they're not native to the area. And one of the issues that we have is that they don't have any natural predators or control. So an invasive that we're dealing with right now, probably all of you have heard about, is the spotted lanternfly, which obviously is not a plant. But one of the big issues is that there's nothing that eats it. And so, I mean, I've trained my children, if they see one, they smash it, which is, I mean, it's terrible. I feel bad, but I'm like, but I also know what it can do to the plants in my yard. And like I said, they can outcompete native plants for lights, nutrient, and water. And another big thing is that they can actually cause changes to the ecosystem. So I don't know if any of you guys have Japanese stiltgrass in your yard. It's a real big problem because I live right by the reservoir. The deer love to spread it. And one of the things that it can do is they actually suck all the nutrients and stuff right from the top of the soil. And so it makes that part not as nutritive, which makes it harder for other things to grow. So garlic mustard's another one. Canadian thistle's another fun one. Okay, let's see what's in the chat. Oh, yes, stiltgrass. Yeah, kudzu, always fun times. So porcelain berry is another fun one. It's stealth. It likes to hide in my bushes. So when do weeds become a problem? So the reason why we're talking about it with dormancy is, so we have a susceptible crop or plant or ecosystem, and weeds love to hang out in what's called the seed bank. And so they can stay dormant for a really long time, especially if they're buried in the soil. But if we do things like construction, or we're working on the roads, or we dig up a plot for a garden, all of a sudden our weeds are close to the soil, they have light, and we have a favorable environment, and they're going to grow. And so they're actually using the ability to be dormant to their advantage. And so what we can see is that mature individuals produce lots of seeds, usually lots of seeds, and we get something called seed rain. I don't know if you guys have ever seen garlic mustard or hairy bittercress. They'll like shoot their seeds. It's pretty impressive. And then we have the surface seed bank or the buried seed bank. And that's the one that we worry most about because it's hard to get rid of something that you can't see. And what we'll see is that a lot of weeds that reproduce by seed actually have really, really small seeds. So a lot of times they're close to the surface, so they can germinate really quickly when temperatures get warm and there's lots of light. And then they can outcompete some of our bigger seeds. So they have small seeds, they have a seed bank. Yeah, and we already talked about dormancy. Okay. So I have one more thing because I think we're supposed to be done at 8, right? Ish. Okay. So that's why, like, you can go into a lot of other things with seed dormancy. So when I was a teenager, we had huge fires in Yellowstone because I grew up in Utah. So it was a big deal because Yellowstone's really, relatively close. You can get there in about 6 hours. And one of the things that happened when we had those forest fires is there were a lot of seeds that needed that fire exposure. So the heat of the fire and then the nutrients released from the fire to germinate. And so, there are other things that can impact whether or not seeds are released from dormancy. I just kind of hit on like the most normal ones. But we're going to end with a fun seed fact. So seed production can also be impacted by the environment that the plant is growing in. So when I was in graduate school, I worked on a plant called Wayule, and that's spelled with a G, is Guayule. So if you've ever read The Grapes of Wrath, they go and they work in the Wayule fields. My father-in-law is the only person who had ever knew what it was used for. So it's used to produce rubber. So Wayule is an interesting plant because it is apomictic. So it can develop seeds without fertilization. So basically you can have, and they're tetraploid So they have 4 copies of all the chromosomes. So they have 72 chromosomes, fun times. And if they don't undergo meiotic reduction, you have an egg cell that has 72 chromosomes in it. It doesn't get fertilized, and you get seeds that are basically clones of the maternal plant. But sometimes you can get meiotic reduction. So you have an egg that has half the chromosome, the original cell of the original plant. It's fertilized by pollen, and you get a hybrid. And you can't tell just by looking at the number of chromosomes. And so when they were trying to breed Waiuli plants during World War II to get rubber, it wasn't working. And that's because they were working with highly apomictic plants. And so as we've learned more about them, what we found is because they grow in the desert, that the closer they are to the maternal plant, the more likely they are to reproduce by apomixis, because that plant already has all the characteristics it needs to survive really well where it is. But as the progeny get further and further away from the maternal plant, they are more likely to undergo sexual reproduction and basically toss the dice and see if they can get progeny that's actually going to be more adapted to the new environment. So we get, and it doesn't necessarily have to do with dormancy, but it's a fun seed fact that I thought I would throw in there, because it's just shows you how wacky and cool different plants are. Okay, so I will stop sharing so that if anybody has any questions, you're free to ask.
Thank you so much. We're going to go into questions and time. Thank you. That was fascinating. It's it's like the hidden life of seeds going on down there. If the seeds, I had one question with when you're talking about the weed seed bank and the role that we play in exposing those seeds. In nature, would that be just other animals digging in the ground, turning things up, or worms, or turning over the soil and things like that?
Yeah, so definitely, if we have burrowing that can expose the weed seed bank. Stilt grass has really, really small seeds, and they can be dormant for up to 7 years. But they're really sticky, and so they can stick to, out by where I live, deer are a real problem. And so they'll just carry them and then just drag them around. So they could also just stay at the surface. And so then they just grow. So there's kind of a couple of different ways. If they're small enough and they don't get buried, they can just chill near the top. But if they've been buried and then get dug up and get exposed to sunlight, they will be like, sweet. And they just grow like crazy. And so a lot of times when you have construction, you'll see a lot of weeds coming up.
And then what is the seeds can stay dormant for quite a while. When does that dormancy, or has that been tested? Is it per species basis of when dormancy, when those seeds are no longer viable, or is it on a spectrum?
It's really, it depends on the type of seed. So like I said, with stilt grass, they think that the seeds are viable for up to 7 years. So I had a real stilt grass invasion in my yard, and so basically I have to use a pre-emergent every spring to make sure that they don't come up, because I don't have the stamina to pull it up by hand all the time. But then other seeds, so I work with a native plant called the Arabidopsis lyrata, and they're usually viable because we've done a lot of germination tests really viable for about 2 years, and then the viability starts to take off. So at about 4 years, you're about 50% viability. So it really depends on the species.
And that viability is based on what is happening in the cell in the seed to lessen viability.
Well, so the embryo is still alive. It's just in a very metabolically inactive state. But if you if you use enough of your resources, then you're going to die. Okay, so Jillian asked, do really tiny dust-like seeds like orchids require a fungal partner to germinate as well as to grow? So some of them do, and I don't know if I can hear stuff from upstairs in my house. I don't know if it transfers down here. So yeah, some seeds are going to have very specific conditions to grow. I know orchids are incredibly hard to grow, and most plants actually do have a fungal symbiont. It's about between 80 to 90% of flowering plants will have some sort of fungal symbiont to reach optimal growth. So it wouldn't surprise me if, especially something like orchids, need something to help them because their endosperm is really tiny. So if they have a fungi that can transport nutrients to them, that's going to be really helpful. Okay, so Anita's asked, why do people do cold stratification for native plants in milk jugs? Is it because they want to plant them in specific places? If you just yeah, if people are stratifying in like milk jugs or in like a plastic bag with like peat moss, then it's usually because they want to plant the seeds in specific places. But if you do just throw your seeds in the yard in the fall, they're going to cold stratify naturally. So there's like a butterfly garden up at our elementary school. They have a bunch of milkweeds there, and the milkweeds just come back every year. And I have a coworker who has a bunch of plants that require cold stratification. She doesn't do anything special. She just lets them spread their seeds and they come up. Oh, how long does it take for an embryo to mature? It really depends on the type of seed. So there's a plant called Arabidopsis thaliana. It's the first plant that they sequence the genome for, and you can go from seed to seed in 6 weeks. So it's really, that's why they use it for genetic studies all the time, because it's super fast. But other seeds are going to take forever. So like pines actually have basically like a 15-month cycle from the time of you make the cone, you pollinate, and it's going to take about 15 months before the seeds are ready to go. So it literally depends on the type of seed. Any other questions?
If you want to raise your hand, we can call, and you want to unmute, I can call on you.
Unknown caller.
That person did not want to ask.
Forget how to raise my hand.
Any other questions?
How do I do?
Oh, Jillian, did you have a question?
All right. Yeah, I had a question.
Okay.
So isn't there a double fertilization thing that goes on with seeds when the female egg more or less is being fertilized? Isn't there sort of a double fertilization where the endospore is produced and then also the embryo or something like that?
Yes. So angiosperms all go through double fertilization. So basically there's 2 sperm cells that are formed by the pollen grain. One fertilizes the egg to make the embryo. The other fertilizes 2 cells that are all 3 cells will fuse together to become the endosperm, which is one of the reasons why the endosperm in angiosperms is usually pretty big comparatively. In pines, they don't have double fertilization, so the endosperm is usually a little bit smaller. But I didn't throw that in here because I was like, well, they said about 45 minutes and I can talk forever. So
thanks. So that's one of the really interesting things, I think.
No problem.
All right. Any other questions for Dr. Blohm?
Okay.
I'm looking for hands. Well, thank you so much. This has been fascinating, giving us an inside peek of things that are going on that we cannot see. And we're glad that you're looking at them. and are able to tell us what's going on. And hopefully, as folks are collecting seeds, they have gotten some tips and tricks that they could use maybe to get their seeds germinated better for the coming year. So thank you very much.
No problem. Thank you so much.
All right, take care, everybody. Stay well, stay curious, stay outside. Bye-bye. We'll see you in a few weeks.
All right.
You're just getting lots of thanks and kudos.
So how do people get on the list to see these lectures, just out of curiosity?
What do you mean get
Like,
on the
how do
list?
people, how do people find out about the lectures? Is it just on your website?
Oh, yeah. Well, we